So welcome to video 5 in our course G06 on transcriptomics. Here we're talking about RNA Seq file formats. So there's different file formats that are associated with these experiments, some of which we do and don't use in our actual practical here. The major ones you might have heard before is called, the first one is called sequence alignment map, which contains essentially a map of our reference genome if our mapping reads to it, and it contains the locations and mapping qualities of those reads mapped to this reference genome or transcriptome, hence the name sequence alignment map. Related to this is BAM, binary sequence alignment map, which is a computer oriented file. It's not human readable, containing a compressed version of the SAM file. These can be indexed as a BAI file that contains a kind of a series of coordinates, and that is essential for reading to other tools such as IGV if you want to view the read pileup of the BAM files themselves. Related to this, then, if we're looking at genomic data, we might have an annotation file available for a reference genome or transcriptome. One standard file format for that is called a GTF file, and there's essentially a map of the annotation for that reference sequence containing the likes of intron and exon boundaries and other features. Similarly, there's also GFF. There's several different types of GFF files, and GFF3 is the most common one that you'll use. And again, these contain annotation in a slightly different format than GTF files, but most tools can use either GTF or GFF. Related to this, we also should be aware that sometimes we need to index our FASTA files just like we index our BAM files, and these contain the coordinates of the different contigs or sequences that are inside our FASTA file. So these FAI files can sometimes are required, and these essentially just contain the contig names, such as chromosome 1 to 10 here, the bases of that sequence, long or small, the index of where that begins in terms of the total sequence there. So you can see this is kind of cumulative instead of this kind of column just containing the lengths, with the number of bases per line and bytes per line as well. So that's a FAI FASTA index file. You should be aware that if you're using read mapping approaches that generate SAM files, these are large and you should delete them after they're generated. You can convert them into BAM files, which are much smaller, and these are more effective for supporting random fast access. You should also note that you can recreate SAM files very easily, as they don't need to be stored. You can then index your BAM files to have get our BAM index file, which will typically have a file ending in .bam.bai. So as you might know from other courses, our SAM file format has a particular set way of arranging the different reads mapped to the reference genome. Typically, there's a large header before we actually get to the real reads, which are down here. These may contain things like the chromosome or contig coordinates and their lengths, a bit like our index file. They may contain information about the reference or tool that's being used. They may contain information about the reads, but the SAM file versions may differ. They may not contain that information. When we go further down to here to actual reads, we can look at the read mapping details for each particular read as we go along. So if we look here at the example of the read ID that's present, and you can actually see the read sequence is listed along here. So there's more than just the sequence information here. We also have the corresponding quality scores of those sequences listed here. And you see that you've unique read ID containing 13M, 17D, 24M at the end. That corresponds to 13 matches, 17 deletions, and 24 matches in this example. You also see that that's the sequence. And you also see here, this is our reposition here, chromosome 1 at base 397, and that's the length 37. We also have the alignment quality as well, 37M, 37 matches. So that's matches right across the whole read length. So typically, we can summarize it that we have our query ID, the alignment flags like 136 here. In this example, the contig name, the position 1 here, the mapping quality, which is typically 60, is what you'll see. The SIGAR format as well, compact idiosyncratic gapped alignment report. the ID of the mate if it's paired, which is here, and then the position of the mate, which is 282 here, and then the Fred scale quality across the whole read, which is there at the end. So there we also can see that we have the sequence listed across here with more base at the end, and then the quality scores corresponding to that do that. And then other things also at the end for when examining sequence quality. So this information is present for each particular read mapped to the reference genome, hence the files tend to be very, very large. To compress them, we turn them into BAM files. So BAM files contain the same information, say about the read name, the location, the alignment, quality scores, the sequence, and the quality scores and other metadata as well, except in a compressed format. So those are SAM BAM files and BAI files. We also have GTF files, and those are just annotation files. And for our purposes here of looking at host gene expression, we might be looking at intron exon boundaries as well. So this means we can identify where the exon junctions are, and we can map unmapped reads to these locations and make sure that our mapping is optimized. So for GTF files, they may have older ones may have slightly different structures, but generally we have a sequence name here, our contig or sequence name, the source. So that's the program that was used to generate it. Here we have our different features that are present. So it might be genes or transcripts or exons. We see the start and end here, and some of these features you'll note overlap. So here we have an exon corresponding to our gene and transcript above. And you see there, and each of these has a ENSG, unique gene identifier, but they also have ENSTs as well, which are unique isoform identifiers as well. You'll also see there's the exon numbers, the gene names, and other, like the transcript version, the gene name version as well, all of which can be used in subsequent analysis. So that kind of concludes a brief introduction into important file formats you may encounter when doing RNA-seq style analysis.